Welcome to the Deep Dive, the show where we cut through the noise, pull out the most important nuggets, and turn complex information into crystal clear insights.
Glad to be here.
Today, we're plunging into the incredible world of nervous and sensory systems.
It really is an amazing topic. I mean, think about it. What actually happens in your brain when you suddenly understand a complex idea or feel a surge of pure joy?
Right. For centuries, understanding something so intricate seemed almost impossible.
Yet, within us, you've got billions of neurons arranged in circuits far more complex than any supercomputer we've built.
Absolutely. And our mission for this deep dive is to explore how animals from the simplest creatures all the way up to us gather, process, and, organize information through these remarkably sophisticated systems.
Yeah, we're going to break down the core concepts from our source material, which is a fantastic chapter from Campbell Biology in Focus.
The idea is to turn what could be pretty dense textbook material into clear, memorable insights for you. Think of it as your shortcut to truly understanding how you perceive the world.
And this isn't just abstract theory. It's about pushing the boundaries of discovery. Take something like rainbow technology, for example.
Oh, yeah, I've heard about that. It sounds incredible.
It is. Scientists can now literally color individual brain cells with different fluorescent proteins, creating these vivid, rainbows.
Wow.
This lets them map neural connections in exquisite detail, giving us unprecedented insight into how information actually moves through specific brain regions. It's completely revolutionizing how we see the brain's wiring.
It's like finally getting a roadmap for the hidden highways of the mind. So, let's unpack this network, starting with the very first steps in its evolution.
Okay.
It's truly wild how much nervous systems have adapted over time. The simplest animals, like hydras, they just have what's called a nerve net, right?
Exactly. A diffuse web of neurons spread throughout their body. It controls basic functions like contracting their gastrovascular cavity. Pretty decentralized, no main command center.
So very simple. But then things started getting more complex.
That's the fascinating part, the evolutionary shift. As animals evolved with elongated, bilaterally symmetric bodies, think flatworms, like planarians, we start to see cephalization.
Cephalization, that's the clustering of neurons at the front end.
Precisely. Sensory neurons and processing centers begin to cluster at the anterior, or front end of the body, forming a primitive brain. It's the beginning of a command center, along with distinct nerve cords running down the body.
Okay, so a head starts to form, essentially.
You could say that, yeah. And as we move up to more complex creatures like insects, their brains become more intricate. They also develop these segmented clusters of neurons along their nerve cords called ganglia.
Ganglia, like mini processing hubs along the way.
Sort of local relay points, yeah. Then, with vertebrates, like us, you get the really distinct central nervous system, or CNS.
That's the brain and spinal cord, the big command center.
Right. And the peripheral nervous system, or PNS, which is all the nerves and ganglia outside the CNS, acts as the communication network.
Carrying messages back and forth.
Constantly carrying information both into and out of that command center, connecting every part of your body.
But here's where it gets really interesting, and something I think people often overlook. Nervous systems aren't just made of neurons.
Oh, absolutely not. They rely heavily on these unsung heroes called glial cells, or glia.
The support crew.
Exactly. Without them, the neurons couldn't function properly. They're incredibly diverse and active. For instance, you have microglia, which are like the brain's immune cells.
Cleaning up debris, fighting off invaders.
Yep. Sweeping up cellular debris and protecting against pathogens. Then you have cells like oligodendrocytes in the CNS and Schwann cells in the PNS.
Ah, the myelin sheath formers.
Right. They wrap nerve fibers in that fatty insulation called myelin. This makes signals race along axons drastically faster. Think of it like upgrading a dirt road to a superhighway for information.
That makes a huge difference in speed.
A massive difference. And then there are the star-shaped astrocytes, which do a huge amount in the CNS.
Okay, what do they do?
Well, they help neurons transfer information, regulate ion concentrations around the neurons, promote blood flow to active neurons, and they even help form the blood-brain barrier.
The protective shield, right?
Yeah.
Carefully controlling what gets into the brain.
Exactly. A vital protective mechanism. And we can't forget radial glia.
Radial glia.
Yeah.
They're involved in development?
Primarily, yes. In developing embryos, they literally form the tracks that new neurons follow as they migrate to their final positions.
Like scaffolding for the developing brain.
A good way to put it. What's even more exciting is that both radial glia and astrocytes can act as stem cells.
Really? They can become other brain cells?
They can self-renew and even generate more specialized brain cells. This opens up incredible potential for future therapies, maybe, to repair damaged brain tissue.
That's incredible, the potential for the brain to heal itself.
Ah,
Okay, so with that amazing support system in place, let's zoom out.
yeah.
How is this complex command center, from its basic highways to its processing centers, actually laid out in vertebrates? Where does the processing begin?
Well, a lot starts at the spinal cord. It's often seen as just a superhighway connecting the brain to the rest of the body, but it's actually smart on its own.
How so?
It generates basic locomotion patterns and, famously, handles those lightning-fast reflexes. Think about touching a hot stove.
Your hand pulls back before you even think about it.
Exactly. Your hand jerks away before your brain even registers the pain signal properly. That's the spinal cord acting independently, prioritizing speed for survival.
So it's like the brain's rapid response system, capable of acting on its own when speed is critical.
Very much so. And if you've ever heard terms like gray matter and white matter,
Yeah, what's the difference there?
gray matter is primarily neuron cell bodies, the processing centers, really, where learning, emotions, a lot of that happens. White matter, on the other hand, consists mainly of bundled axons, often myelinated ones. It's the wiring linking the CNS to the PNS and allowing signals to fly between different brain regions.
And their location differs, right, between the brain and spinal cord.
It does. In the spinal cord, the white matter forms the outer layer, the communication lines, while the gray matter is more butterfly-shaped in the center. In the brain, it's kind of reversed. Gray matter forms the outer cortex and some deep nuclei, while white matter is mostly in the interior, connecting everything.
Got it. And there's fluid involved, too.
Right. The CNS has these critical fluid-filled spaces, the central canal deep within the spinal cord, and interconnected ventricles within the brain. They're filled with cerebrospinal fluid, or CSF.
CSF, what does that do?
It's formed from arterial blood, and it basically bathes the CNS. It delivers nutrients and hormones, cushions the brain, and importantly, carries away waste products, like the brain's own personal circulatory and waste disposal system.
Okay, so that covers the CNS basics. Now, moving to the peripheral nervous system, the PNS, that's the bridge, connecting the CNS to everything else.
Exactly. You have afferent neuron.
Arrival, information coming in.
Good way to remember it. They're like information scouts, carrying sensory signals from the body toward the CNS. Then there are efferent neurons.
Efferent, exit, commands going out.
Perfect. They're like command couriers, carrying instructions away from the CNS to muscles and glands. Most nerves are actually bundles containing both afferent and efferent fibers working in a two-way flow.
Makes sense. And the PNS has different parts too, right?
It does. The efferent component further divides. First, there's the motor system, which controls your skeletal muscles.
Like deciding to wave your hand.
That's the voluntary part. But it also includes involuntary control, like those spinal reflexes we talked about.
Okay. And the other part?
That's the autonomic nervous system. This generally operates without you consciously thinking about it. It controls things like your heart rate, digestion, breathing, gland secretion, all that background stuff.
The automatic stuff.
Pretty much. And the autonomic system itself has two main divisions, often with opposing effects. There's the sympathetic division.
That's the fight or flight response.
Exactly. It ramps up your heart rate, slows digestion, diverts blood to muscles, releases adrenaline, gets you ready for action when you're under stress or perceive a threat.
And the opposite.
Is the parasympathetic division. Think rest and digest. It generally has calming effects, slows your heart rate, stimulates digestion, promotes self-maintenance activities. They often work in balance.
Fight or flight, or rest and digest. Got it.
And there's actually a fascinating third component, sometimes considered part of the autonomic system, the enteric nervous system.
Enteric, like in the gut.
Precisely. It's a distinct network of neurons located entirely within the walls of your digestive tract, pancreas, and gallbladder. It controls digestive functions directly and somewhat independently from the brain and spinal cord. It's sometimes called the second brain.
Wow, a whole neural network just for digestion. Okay, so we've built the network. Now, how do we actually understand what's happening inside the brain's master control center when it processes all this information? How do scientists peek inside?
That's a huge question, and modern functional imaging techniques have been revolutionary. Things like functional magnetic resonance imaging, or fMRI.
fMRI, how does that work?
In simple terms, it detects changes in blood flow in the brain. When a brain area becomes more active, it needs more oxygen, so blood flow increases to that area. fMRI picks up on these changes, creating maps of brain activity.
So you can see which parts light up during certain tasks.
Exactly. I remember seeing a great study using fMRI. Researchers mapped brain activity while people listened to music they described as either sad or happy.
Yeah, and what did they find?
The results were striking. Sad music significantly increased activity in the amygdala, a region we know is involved in processing emotions, especially negative ones and emotional memory.
Okay.
But happy music lit up a different area, the nucleus accumbens, which is a key part of the brain's reward and pleasure circuits.
So you could literally see different emotions activating different brain regions.
It's a beautiful illustration of the brain's regional specialization. And these techniques aren't just for research, they have real-world applications now.
Like what?
Monitoring recovery after a stroke, mapping abnormalities in conditions like migraines, even guiding surgeons during brain operations to avoid damaging critical areas. They give us invaluable insights.
Incredible tools. So let's take a quick tour of the main parts of the vertebrate brain. It starts simple in the embryo, right?
Yeah, it begins with three basic bulges, the forebrain, midbrain, and hindbrain, which then differentiate and grow into all the complex adult structures.
Okay, starting at the base.
As the base connecting to the spinal cord, you have the brainstem. This includes the midbrain, the pons, and the medulla oblongata.
The brainstem. What's its main job?
Think of it as the brain's essential life support and relay station. It handles absolutely critical functions you don't consciously control: breathing, heart rate, digestion, maintaining consciousness, arousal, sleep cycles. It also relays information between the rest of the brain and the spinal cord.
And reflexes, too.
Yes, it coordinates certain automatic reflexes, like visually tracking objects. Interestingly, most axons carrying movement instructions cross over from one side of the brain to the other side of the body within the medulla.
Ah, so that's why the right side of the brain controls the left side of the body and vice versa.
That's largely where it happens. Now, tucked behind the brainstem is the cerebellum.
The cerebellum.
Yeah.
Movement and balance, right?
Primarily, yes. It's absolutely vital for coordinating movement, maintaining balance, and ensuring fine motor skills are smooth and precise. But it's also surprisingly involved in learning and remembering motor tasks. Think about learning to ride a bike or play an instrument.
Damage there would make coordination really difficult.
Extremely erratic, yes. Moving up, we find the diencephalon, nestled between the brainstem and the cerebrum. It contains key structures like the thalamus.
The thalamus, the sensory switchboard.
That's a great analogy. Almost all sensory information heading to the cerebrum site, sound, touch, taste, first passes through the thalamus. It acts like a relay station, sorting that information and sending it to the appropriate processing centers in the higher brain regions.
Okay, so it directs the traffic. What else is in the diencephalon?
A tiny but incredibly powerful structure called the hypothalamus, located just below the thalamus.
Hypothalamus. That controls homeostasis, right? Body temperature.
Exactly. It's your body's internal thermostat and a central command center for maintaining homeostasis. It regulates hunger, thirst, body temperature, circadian rhythms, and even initiates your fight or flight response, partly through its control over the pituitary gland.
And it has that internal clock component.
Yes, it houses the suprachiasmatic nucleus, or SCN, which is the master pacemaker for our biological clocks. We'll touch on that more. There's also the epithalamus, which includes the pineal gland that produces melatonin.
Melatonin for sleep.
Okay.
And then the biggest part.
The cerebrum, the large, highly folded outer part of the brain. This is the center for higher level functions. Learning, memory, emotion, perception, voluntary movement, consciousness itself.
Our thinking cap.
Essentially, yes, it's divided into the right and left cerebral hemispheres.
Connected by.
A thick band of nerve fibers called the corpus callosum, which allows the two hemispheres to communicate constantly.
Okay, let's circle back to those rhythms you mentioned: arousal, sleep, and the biological clock. Sleep isn't just switching off, is it?
Not at all. Sleep is a remarkably active state for the brain. We know this from looking at brainwave patterns using an electroencephalogram, or EEG. There are distinct stages of sleep, each with characteristic brain activity.
And these cycles are driven internally.
Largely, yes. They're a prime example of circadian rhythms, these roughly 24-hour cycles of biological activity. They're driven by an internal molecular clock mechanism.
So even without daylight cues.
Even if you were kept in constant darkness, your internal clock would likely maintain a cycle of about 24.2 hours, pretty close to the actual day length.
And the SCN in the hypothalamus is the key.
It's the master pacemaker. It receives light input from your eyes, which helps synchronize your internal clock with the external light-dark cycle. It keeps everything aligned.
That's fascinating. I heard dolphins do something weird with sleep.
They do. Dolphins can sleep with only one hemisphere of their brain at a time.
Wow. Why?
It allows them to continue swimming, surface to breathe, and remain partially alert to their surroundings even while getting rest. An incredible adaptation.
Truly amazing. Now, what about emotions? Where do they fit in?
Emotions aren't generated by just one single structure, but a network of brain regions often referred to as the limbic system.
The limbic system, where's that?
It's sort of a border region around the brainstem involving structures like the amygdala, the hippocampus, and parts of the thalamus and hypothalamus.
And the amygdala is key for emotional memory.
Very much so. It links emotions, particularly fear, to specific memories. It's why recalling a frightening event can trigger physiological responses, like a fast heart rate or sweating, even long after the danger is gone. The limbic system essentially attaches emotional feelings to basic survival functions.
Makes sense. And related to that is the brain's reward system.
Absolutely. There's a specific neural circuit that motivates behaviors essential for survival and reproduction, things like eating, drinking, sex. It makes these activities feel rewarding or pleasurable.
How does that work? Dopamine is involved, right?
Yes, dopamine is a key neurotransmitter here. Neurons originating in an area called the ventral tegmental area, or VTA, release dopamine onto neurons in regions like the prefrontal cortex and, importantly, the nucleus accumbens. This dopamine signal essentially tells your brain, that was good, remember it, do it again.
Which is great for survival, but it can be hijacked.
Precisely. This is where drug addiction comes in. Many addictive substances, alcohol, cocaine, nicotine, heroin, work by artificially hijacking and massively enhancing the activity of this dopamine pathway. They do it in different ways. Cocaine, for example, blocks the reuptake of dopamine from the synapse, meaning the dopamine hangs around longer, intensifying the reward signal. Nicotine directly stimulates those dopamine-releasing VTA neurons.
So it creates an unnaturally strong reward.
Exactly. And with repeated use, addiction can develop. This often involves long-lasting changes in the reward circuitry itself. The brain adapts to the drug, leading to intense cravings and compulsive drug-seeking behavior, often independent of any pleasure the drug still provides.
It rewires the motivation system.
In a very powerful and often detrimental way. Animal studies show this vividly. Rats will self-administer drugs like cocaine compulsively, even choosing the drug over food, sometimes to the point of starvation. It highlights the loss of control involved.
A stark illustration. Okay, let's shift focus now to the thinking brain, the cerebral cortex, and its role in cognition, memory, and our amazing adaptability.
Right, the cerebral cortex. This outer layer of the cerebrum is absolutely essential for the functions we consider uniquely human, like complex language, abstract thought, consciousness, and awareness.
And it's organized into different functional areas.
Yes. Broadly, you have sensory areas that receive incoming information, motor areas that send out commands for movement, and large association areas that integrate information from multiple sources, allowing for complex thought.
And those four lobes we hear about.
The frontal, parietal, temporal, and occipital lobes. Each is generally associated with different primary functions, although there's a lot of integration. For example, the occipital lobe is primarily for vision, the temporal lobe for hearing and language comprehension.
Speaking of language, the discoveries about Broca's and Wernicke's areas were huge, weren't they?
Foundational. Doctors in the 19th century linked specific language problems to damage in particular brain regions. Damage to Broca's area, usually in the left frontal lobe.
And people could understand language, but couldn't speak fluently.
Exactly. They knew what they wanted to say, but struggled to form the words. Then, Carl Wernicke identified Wernicke's area, typically in the left temporal lobe.
And damage there affected understanding.
Right. Patients could often speak, sometimes fluently, but their words might not make sense, and crucially, they couldn't comprehend language spoken or written.
This points to that idea of lateralization, right? Hemisphere specializing.
Yes. For most people, the left hemisphere is dominant for language, math, and logical processing. The right hemisphere tends to be more involved in spatial reasoning, face recognition, musical ability, and interpreting emotional tone.
But they don't work in isolation. That corpus callosum connects them.
Critically,
Yeah.
it allows constant communication. There have been cases where the corpus callosum was surgically severed, maybe to treat severe epilepsy.
Split brain patients.
Exactly. And studying them revealed fascinating things. For instance, if you showed an object to their left visual field only, which sends info to the right hemisphere.
They couldn't name the object.
Correct, because the language centers are typically in the left hemisphere, and the information couldn't cross over. However, they could often pick out the object by touch with their left hand, which is controlled by the right hemisphere. It's like two separate spheres of awareness.
Mind-boggling. So how does information actually flow through the cortex during processing?
Well, sensory input from your eyes, ears, skin via those somatosensory receptors for touch, pain, temperature generally gets routed first by the thalamus to the primary sensory area for that sense in the cortex.
Okay, initial reception.
Then the information moves to nearby association areas for more complex processing and integration. So your primary visual cortex registers lines and edges, but the visual association area helps you recognize that pattern as a specific object, like a face.
Putting the pieces together.
Exactly. From there, information often converges on the prefrontal cortex, the very front part of the frontal lobe. This area is crucial for planning, decision-making, working memory, what we call executive functions. It formulates a plan for action.
And then sends commands to the motor areas.
Right, which then execute the movement.
The prefrontal cortex and its role in personality. The Phineas Gage story is always mentioned here.
It's a classic tragic case. In 1848, an accidental explosion sent an iron rod straight through Gage's frontal lobe. Miraculously, he survived.
But he wasn't the same person.
Not at all. Intellectually, he seems largely intact, but his personality changed dramatically. He became impatient, profane, emotionally detached, unable to stick to plans. It was one of the first key pieces of evidence linking the prefrontal cortex specifically to personality, temperament, and decision-making.
Such a powerful example. Now, thinking about evolution, how did complex cognition evolve? We see it in primates and cetaceans with their big, folded cortexes.
Right, that highly convoluted cerebral cortex making up about 80% of the human brain's mass was long thought to be the key prerequisite for higher intelligence.
But then we learned about birds, right? Challenging the bird brain idea.
Absolutely. That old idea that birds lack intelligence because they don't have that folded cortex has been completely overturned. We now know birds can be incredibly smart.
Like remembering where they hid food.
Exactly. Western scrub jays, for example, remember not just where they cached food, but also when they cached it and who might have been watching them. African gray parrots can understand abstract concepts like same, different, even none.
So how do they manage that without the folded cortex?
It turns out they have a different brain organization. Instead of a layered cortex like ours, they have a densely packed nuclear organization of neurons in a structure called the pallium. Evolution essentially found two different architectural solutions to achieve complex cognitive function.
Different paths to intelligence. That's fascinating. And underpinning learning and adaptation is this concept of neuronal plasticity.
Yes, a fundamental property of the nervous system. Neuronal plasticity is the ability of the nervous system to remodel itself structurally and functionally in response to its own activity, to experience.
It's happening where?
Primarily at the synapses, the junctions where neurons communicate. Connections can be strengthened if they're used effectively, weakened if they're not, and new connections can form while others are pruned away.
Like optimizing the network based on traffic.
That's a good way to think about it. If signals flowing through a particular pathway lead to a useful outcome, that pathway gets reinforced, making future signaling easier or more effective. It's like widening a highway ramp that gets a lot of important traffic.
And this is crucial for learning and memory.
Absolutely fundamental. Learning involves modifying those synaptic connections. In fact, problems with neuronal plasticity are thought to underlie some neurological and developmental disorders, like potentially autism spectrum disorder, where activity-dependent synaptic remodeling might be disrupted.
So plasticity is key for both normal function and potentially for disease. How does it relate specifically to memory?
Memory formation is essentially a process of synaptic plasticity. We often distinguish between short-term memory and long-term memory.
Short-term is temporary.
Right. It might involve temporary changes in synaptic strength or activity patterns, often involving the hippocampus. To form a long-term memory, those changes need to become more stable and structural, often involving the transfer of information from the hippocampus to be stored more permanently within the cerebral cortex itself.
Making the connections more robust.
Exactly. This process of transferring information from short-term to long-term storage is called memory consolidation, and it's thought to happen significantly during sleep.
Ah, so that's why sleep is important for learning.
It seems to be crucial. During sleep, the hippocampus might replay patterns of activity from the day, helping to strengthen the corresponding connections in the cortex. Some researchers even think this hippocampal reactivation might form the basis of some of our dreams.
Wow. And the evolutionary advantage is integrating new stuff with old stuff.
Precisely. Gradually integrating new knowledge into your existing stores of information allows you to form more meaningful associations and build a richer understanding. It's why learning gets easier when you can connect new facts to things you already know.
Makes sense.
Hmm.
And scientists are still pushing to understand all this.
Massively. Huge initiatives like the Brain Initiative are aiming to map brain circuits in unprecedented detail, measure their activity patterns, and ultimately figure out how all this neural activity translates into actual thoughts, feelings, and behaviors. It's a huge frontier.
Absolutely.
Mm.
Okay, let's shift gears one last time to sensing the world. How do we actually convert external stimuli, light, sound, touch, into something our brain understands?
It all follows a basic sensory pathway. Step one is sensory reception.
Detecting the stimulus.
Right. Specialized sensory cells, which can be neurons or other cell types, detect a specific type of stimulus. And they can be incredibly sensitive, like photoreceptors in your eye detecting a single photon of light.
Amazing. What's next?
Step two, sensory transduction. This is the crucial conversion step. The sensory cell converts the energy of the stimulus light, energy, mechanical pressure, chemical energy into an electrical signal. Specifically, a change in the cell's membrane potential called a receptor potential.
So physical energy becomes electrical energy.
In essence, yes. Step three is transmission. That receptor potential, which is graded, its size varies with stimulus strength, influences the generation of action potentials, those all-or-none nerve impulses that travel along axons to the CNS.
And stronger stimuli mean more action potentials.
Generally, yes. The intensity of the stimulus is typically coded by the frequency of action potentials. A louder sound or brighter light triggers action potentials more often.
Okay, reception, transduction, transmission.
Sure.
And finally,
Step four, perception. This happens in the brain. The brain receives these incoming action potentials, processes the information, and interprets it, constructing our conscious perception, seeing colors, hearing sounds, feeling textures.
so the perception is created by the brain, the old tree falling in the forest question.
Exactly. If no one or no animal with an auditory system is there to detect the sound waves and transduce them into nerve signals that a brain can interpret, was there really a sound? Philosophically debatable, but biologically, the perception of sound happens in the brain.
And how does the brain know if it's getting a light signal or a sound signal if the action potentials are similar?
It's all about the wiring. Action potentials arriving via the optic nerve are interpreted as light because they land in the visual cortex. Action potentials arriving via the auditory nerve are interpreted as sound because they land in the auditory cortex. The pathway defines the perception.
Got it. And our senses can adjust, right? Amplify things.
Yes. Amplification can occur, strengthening the sensory signal during transduction. The tiny bones in your middle ear, for instance, amplify the pressure of sound waves significantly before they reach the inner ear.
And we also adapt,
Hmm.
like not noticing your clothes after a while.
That's sensory adaptation, a decrease in responsiveness to a constant stimulus. It's incredibly important. It prevents your nervous system from being overwhelmed by unchanging information, allowing you to focus on detecting changes in your environment, which are often more significant.
Makes sense. Okay, let's quickly run through the main types of sensory receptors, 5 categories.
That's right. First, mechanoreceptors. These respond to physical deformation, touch, pressure, stretch, motion, sound. Things like the sensitive receptors at the base of a cat's whiskers or the hair cells in our ears.
Okay.
Second, electromagnetic receptors. These detect forms of electromagnetic energy. The most familiar is light, detected by photoreceptors in our eyes. But some animals have amazing abilities here.
Like detecting electric fields.
Yeah, platypuses use receptors in their bills to detect the weak electric fields generated by prey underwater. Some fish generate their own electric fields. And many migrating animals, like birds, butterflies, even whales, can sense the Earth's magnetic field for navigation, likely using specialized proteins or tiny magnetic particles.
Incredible. Third type.
Thermoreceptors. These detect heat and cold. You have them in your skin, obviously, but also internally, like in the hypothalamus, to monitor core body temperature.
And the spicy food thing.
Ah, yes. Capsaicin, the chemical in chili peppers, activates the same receptor that responds to painfully high temperatures, above 42 degrees C or 108 degrees there. That's why it feels hot. Similarly, menthol activates receptors normally triggered by cold temperatures, below 28 degrees C or 82 degrees there, creating that cooling sensation.
So it tricks the temperature sensor. Fourth type.
Pain receptors, or more technically, nociceptors. They respond to noxious stimuli, things that could cause tissue damage, like extreme pressure, extreme temperatures, or certain chemicals released by damaged tissues. They trigger protective reflexes and the perception of pain.
And things like aspirin work on these pathways.
Indirectly, yes. Damaged tissues release chemicals like prostaglandins, which sensitize nociceptors, making them respond more readily. Aspirin and ibuprofen inhibit the synthesis of prostaglandins, thus reducing pain signaling. They act as analgesics.
Okay, and the final category.
Chemoreceptors. These respond to chemicals. Some are general, detecting overall solute concentration, like the osmoreceptors in your brain that trigger thirst when your blood gets that concentrated.
And others are specific.
Very specific. Receptors for glucose, oxygen, carbon dioxide, amino acids. And of course, this category includes our senses of smell, olfaction, and taste. Gustation.
Smell detecting airborne chemicals.
Right, odorants. Humans have around 400 different types of olfactory receptor proteins, allowing us to distinguish potentially thousands of different smells.
Yeah.
A Nobel Prize was awarded for figuring that out.
And taste detects chemicals in solution. Pastants.
Exactly. We recognize 5 primary tastes.
Right.
Sweet, sour, salty, bitter, and umami, a savory taste associated with glutamate like in MSG or aged cheese.
And the tongue map is wrong.
Mostly, yes. That old map showing specific regions for each taste is largely a myth. Any region of the tongue with taste buds can detect all 5 tastes, although individual taste cells within a taste bud are typically specialized for just one type of tasting.
Good to know. Okay, let's dive into 2 senses that rely heavily on those mechanoreceptors, hearing and balance.
Right, both depend on specialized mechanoreceptor cells, hair cells detecting the movement of fluid or tiny particles.
How do simpler animals manage balance?
Many invertebrates use organs called statocysts. These are chambers lined with ciliated receptor cells, containing dense granules called statoliths.
Like little pebbles.
Essentially, yes. Gravity pulls the statoliths down, and as the animal moves, the statoliths shift, stimulating different receptor cells, providing information about the animal's orientation relative to gravity.
Clever. And hearing in invertebrates.
Varies a lot. Insects might use vibrating body hairs or specialized tympanic membranes, kind of like eardrums, often located on their legs or abdomen.
Okay, now for the human ear, it's complex. Let's start with hearing.
It's a journey for sound waves. First, the outer ear, the pinna and auditory canal, collects the waves and channels them to the tympanic membrane, or eardrum.
The eardrum vibrates.
Right. Those vibrations are then transferred across the middle ear by 3 tiny bones, the malleus, impulpicus, and stapes, hammer, anvil, and stirrup. These bones act like levers, amplifying the vibrations.
Amplifying the sound.
Significantly. The stapes then pushes against a membrane called the oval window, transmitting the vibrations into the fluid-filled cochlea in the inner ear.
Cochlea. The snail-shaped thing.
That's the one. Inside the coiled cochlea, the pressure waves created in the fluid cause a structure called the basilar membrane to vibrate. Sitting on the basilar membrane are the crucial hair cells.
The mechanoreceptors.
Exactly. As the basilar membrane vibrates, the hairs on these cells bend against an overlying membrane called the tectorial membrane. This bending physically opens or closes ion channels in the hair cells.
Triggering the signal.
Yes, changing the membrane potential and causing the release of neurotransmitters, which generates action potentials in the auditory nerve that travel to the brain. Your brain interprets these signals as sound.
How does it know volume and pitch?
Volume, or loudness, corresponds to the amplitude of the sound wave. A louder sound causes more vigorous vibrations, bending the hair cells more, leading to more frequent action potentials.
Okay, and pitch?
Pitch, or frequency, is encoded by where along the basilar membrane the vibrations are strongest. The membrane is tuned stiffer and narrower near the oval window, responding to high frequencies, and wider and more flexible at the far end, responding to low frequencies. The brain knows the pitch based on which hair cells are sending signals.
A place code for frequency. Amazing. Now, what about balance or equilibrium? Also in the inner ear.
Yes, connected to the cochlea, we have 2 main sets of structures. First, the utricle and saccule.
What do they detect?
They sense your head's position relative to gravity, are you upright or tilted, and linear acceleration, like when you start moving forward in a car.
How? More hair cells.
Yes, but these hair cells have their hairs embedded in a gelatinous material containing tiny calcium carbonate crystals called otoliths, literally ear stones.
Otoliths, like the stadoliths in invertebrates.
Very similar concept. When you tilt your head or accelerate, gravity or inertia causes the dense otoliths to shift, lagging behind slightly. This bends the hairs of the hair cells, sending signals to your brain about the direction of tilt or linear motion.
Okay, so that's for linear movement and gravity. What about rotation? Spinning.
That's the job of the three semicircular canals. They're arranged in three different planes, roughly at 90-degree angles to each other, corresponding to the three dimensions of space.
How do they work?
Each canal is filled with fluid. At the base of each canal, there's a swelling containing a cluster of hair cells with their hairs embedded in a gelatinous cap called a cupula. When you turn your head, the fluid inside the corresponding canal lags behind due to inertia, pushing against the cupula and bending the hair cells.
Signaling rotation in that plane.
Exactly. The three canals together can detect rotation in any direction, and that dizziness you feel after spinning. That's because the fluid in the canals continues to swirl for a bit even after you've stopped, still stimulating the hair cells and telling your brain you're moving, even though your eyes say you're not. That conflict causes the dizzy feeling.
Ah, that makes sense. Okay, last major sense. Vision. Seeing the light.
A sense central to most animals. And what's fascinating is that despite the incredible diversity of eye types out there, the basic light-capturing molecule is remarkably conserved across the animal kingdom.
Suggesting a common evolutionary origin.
Strongly suggesting it, yes.
Yeah.
At least for the photoreceptor mechanism itself.
So what are some of the different eye types?
Well, at the simple end, you have things like the ocelli or eye spots of planarians. They don't form images, but just detect light intensity and direction, helping the planarian move away from light towards safer, darker areas.
Simple light detection.
Then you have compound eyes, famously found in insects and crustaceans. These are made up of thousands of individual light-detecting units called ommatidia, each with its own lens and photoreceptor cells.
Like thousands of tiny eyes working together.
Kind of. Each ommatidium samples a small part of the visual field. Compound eyes aren't great for sharp detail, but they are exceptionally good at detecting movement, a huge advantage for flying insects or detecting predators.
Okay. And the eyes like ours.
Those are single-lens eyes, also found in spiders, some mollusks like squid and octopuses, and all vertebrates. They work much like a camera.
How so?
Light enters through a transparent outer layer, the cornea, then passes through an adjustable aperture, the pupil, controlled by the iris, and is focused by a single lens onto a layer of photoreceptor cells at the back, the retina.
And the lens focuses the light.
Yes. In mammals like us, muscles change the shape of the lens to focus on objects at different distances.
Okay, the retina at the back contains the photoreceptors, rods and cones.
Exactly. Rods are highly sensitive to light, responsible for our vision in dim conditions, essentially black and white or grayscale night vision. Cones are less sensitive, but provide color vision.
And we have different types of cones.
Humans typically have three types of cones, each most sensitive to different wavelengths of light, broadly corresponding to red, green, and blue. The brain compares the signals from these three cone types to perceive a whole spectrum of colors.
And what's inside rods and cones that actually detects the light?
They both contain a light-absorbing molecule called retinol, which is actually a derivative of vitamin A. This retinol molecule is bound to a protein called an opsin. The specific type of opsin determines which wavelengths of light the pigment absorbs best. In rods, the pigment is called rhodopsin.
Okay, retinol plus opsin. So what happens when light hits it?
This is the key transduction step. Light causes the retinol molecule to change its shape it isomerizes, flipping from a bent cis form to a straight trans form.
Just a shape change.
Yes,
Yeah.
but this shape change triggers a cascade of events within the photoreceptor cell. The activated opsin activates a G protein, which in turn activates an enzyme called phosphodiesterase.
A molecular chain reaction.
Exactly, and this enzyme breaks down another molecule called cyclic GMP, or cGMP. Now here's the slightly counterintuitive part.
Okay.
In the dark, cGMP levels are high, and this keeps sodium channels open in the photoreceptor membrane. So the cell is actually depolarized relatively positive inside and continuously releases a neurotransmitter called glutamate.
So in the dark, it's on, releasing signals.
Right. But when light hits rhodopsin and triggers that cascade, cGMP levels fall. This causes those sodium channels to close.
So the cell becomes more negative inside.
It hyperpolarizes, yes, and this hyperpolarization stops the release of glutamate. It's this decrease in glutamate release that signals to the next neurons in the retina that light has been detected.
Wow, so light turns the signal off rather than on.
For the photoreceptor itself, yes. It's a fascinating system. Then enzymes convert the retinal back to its cis form, resetting the system, ready to detect more light.
Did this explain being temporarily blinded by bright light?
It does. When you go from bright sunlight into a dark room, your rods have been overwhelmed. So much rhodopsin has been activated, we call it bleached, that it takes a few minutes for enough of it to reset before the rods become sensitive enough to detect the dim light. That's the period of dark adaptation.
Ah, makes sense. So the retina does more than just detect light.
Oh yes, significant processing happens right there. Rods and cones synapse with bipolar cells, which then synapse with ganglion cells. Other neurons, called horizontal cells and amacrine cells, integrate information laterally across the retina, helping to sharpen contrast and detect edges.
So it's already starting to analyze the image.
Definitely. Each ganglion cell receives input from a number of photoreceptors covering a small area of the retina, its receptive field. The smaller the receptive field, the finer the detail the ganglion cell can resolve.
And the ganglion cell axons form the optic nerve.
Exactly. The axons bundle together to form the optic nerve, which carries the process signals out of the back of the eye towards the brain.
And the crossover.
At the optic chiasm, yes. Axons originating from the inner half of each retina, receiving light from the opposite visual field, crossover. The result is that signals from your left visual field, seen by parts of both eyes, are processed in the right hemisphere of your brain, and signals from the right visual field go to the left hemisphere.
That crossover again.
It's a common theme in vertebrate wiring. From the thalamus, visual information travels to the primary visual cortex in the occipital lobe, and then to numerous other cortical areas for further processing.
How much the cortex is involved in vision?
A huge amount. Estimates suggest at least 30% of the human cerebral cortex is involved in some aspect of visual processing, analyzing color, motion, depth, shape, recognizing objects and faces. It's a massive computational task.
Incredible. And color vision relies on those three cone types.
Primarily, yes.
How?
Each cone type has a slightly different opsin protein called photopsins, making them sensitive to different ranges of wavelengths, red, green, or blue. The relative activity of these three cone types allows us to perceive millions of colors.
And color blindness.
Often results from genetic mutations affecting one or more of the photopsin genes. Since these genes are often located on the X chromosome, color blindness is much more common in males.
Makes sense. And animals active at night.
Nocturnal mammals, like cats or owls, typically have retinas dominated by rods, giving them excellent night vision but usually much poorer color vision compared to us diurnal creatures.
And why is it easier to see a dim star if you look slightly away from it?
Because the very center of your retina, the fovea, where your vision is sharpest in bright light, is packed almost exclusively with cones for detailed color vision. There are very few rods right there, but just outside the fovea, the density of rods increases significantly. So, by looking slightly to the side, you focus the dim starlight onto an area richer in those highly sensitive rods, making it easier to detect.
Clever trick. What an incredible journey through our nervous and sensory systems. It's amazing how it all connects.
It really is. We've seen how these complex command and control centers, from the simplest nerve nets all the way to our intricate brains, rely on these complex circuits of neurons and their absolutely essential glial cell support team.
And how our sensory systems perform this almost magical feat of transduction, turning physical and chemical stimuli from the world around us and inside us
Yes.
into the electrical language of the nervous system.
Which the brain then processes and interprets, constructing our perceptions, our thoughts, our entire conscious experience. Features like regional specialization in the brain, the incredible plasticity of neuronal connections, and countless evolutionary adaptations allow for such diverse and sophisticated functions.
It truly shapes our reality.
Yeah.
Which leads to a final thought.
Yeah.
Considering that our entire experience of the world, every sight, sound, feeling, thought, is ultimately a construction created by the intricate activity within our brain, based on the signals it receives. What does that truly imply about the nature of reality itself? And maybe even the potential for forms of perception or understanding that our current biology doesn't allow us to even imagine.
That is definitely something to ponder. The limits of our own perception defined by the very systems we've been discussing.
Absolutely. Well, thank you for joining us on this deep dive into the nervous and sensory systems. We hope you gained some memorable insights that maybe shift how you see your own remarkable biology.